Good afternoon. Myself, Rahul Chavhan, Assistant Professor, Vilasrao Deshmukh College of Agricultural Biotechnology, Latur. Here I am going to present my topic biotechnological approaches to engineer virus resistance in crop plants. As we know, the virus diseases are the obligate intracellular parasites that do not have their molecular machinery to replicate without host. So they required a host machinery for their replications and survival into the host. So Mayers in 1886 they worked on tobacco mosaic virus very first time. Later on, Bajering in 1898 proposed the term contagium vivum fluidum. So this is the first term given to the virus. So later, Mackey and Warner in 1933, so they discovered tulip breakings which are associated with virus disease, and there are several components through which virus body is made. So it includes protein, nucleic acid, cations, polyamines, enzymes, lipids, and water. So why there is requirement or need for engineering resistance against plant viruses? Because viruses are the major and important constraints that threaten agricultural developments and global food security. Viral diseases globally reduces crop yields to the extent of 20 to 40%, and there are no availability of resistant genes into the cultivated species. So most of the cultivated species are being susceptible to viral diseases, and viruses causes devastating losses because of their systemic infections. So they spread rapidly and infect to the plants, so entire fields may get infected by these viruses. Viral epidemics due to emergence of new strains of virus or a new form of vectors can cause devastating losses, and they are very difficult to identify because of their synergistic or antagonistic effects between different strains of viruses on the same crop plant. So they are vary in their genetic makeup and always change their mode of transmission and disease symptoms. So it is very difficult to identify and implement a management strategies. So that's why it is very crucial and becoming a need to engineer resistance against these viruses by using biotechnological intervention. So what are the biotechnological interventions through which we can attempt virus resistance? Okay, program. So first one is genetic engineering. So genetic alteration of the plant carrying the foreign or viral genes inducing resistance against challenge viruses. There are three important strategies for engineering virus resistance. So first one is host plant resistant gene, for example R gene, uh, which can be introduced into the susceptible host. Then pathogen derived resistance, for example, CP, that is coat protein, movement protein, and replicase protein genes we can incorporate into the host plant. And third one is pathogen targeted resistance. So, for example, proteins or enzymes that specifically target viral components, example, TALEN, CRISPR, Cas9, and zinc finger nuclease. So, to the conclusions, so there is need to establish safe, eco-friendly, and novel approaches to handle viruses in plants, to develop and modify novel pathways in plants to target broad range of plant viruses, utilize CRISPR-Cas9, TALEN, and zinc finger nuclease to provide resistance against multiple DNA and RNA viruses simultaneously, and stable genome-edited transgenic plants blended with plant breeding programs can incorporate viral resistance into a high-yielding commercial hybrids or varieties. These are the references which we have utilized for preparation of this presentation. Thank you so much.